By examining the cells under a microscope, we can observe both animal and plant cells closely. The micrographic images of animal cells typically appear round or irregular in shape, with a distinct nucleus. In contrast, the images of plant cells reveal a rectangular shape with a cell wall. The nucleus is visible within these cells, and chloroplasts may also be present, which are essential for photosynthesis. Let's explore the differences between animal and plant cells by examining their structures more closely, one at a time. This is how the structure of an individual cell appears. By examining it closely, we can identify the similarities and differences between animal and plant cells. The general diagram of an animal cell illustrates a typical animal cell with various organelles, including the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and cell membrane. In comparison, the general diagram of a plant cell shows organelles such as the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuole, chloroplasts, and cell wall. In this section, we will concentrate on specification number 3, which states: identify on diagrams, photomicrographs, or electron micrographs the ribosomes, mitochondria, nucleus, cytoplasm, and cell membrane in an animal cell. This specification requires us to examine the basic structure of animal cells and their organelles. By carefully studying a labeled diagram, we can identify several organelles, such as the endoplasmic reticulum, Golgi bodies, peroxisomes, microtubules, microfilaments, centrioles, centrosome matrix, lysosomes, and microvilli. However, in accordance with the OLevel syllabus, we will focus specifically on ribosomes, mitochondria, nucleus, cytoplasm, and cell membrane in an animal cell. Along with the diagram, we will also examine photomicrographs and electron micrographs to identify these structures. In the examined photomicrograph of an animal cell, several key structures are identifiable. The cytoplasm, a jelly-like substance, fills the cell and facilitates various cellular processes. The nucleus, a prominent dark round structure, acts as the control center for the cell. While mitochondria are small and not distinctly visible due to the current magnification, higher magnification would allow us to observe these essential organelles involved in energy production. It's noteworthy that ribosomes cannot be seen with a light microscope, reflecting their minuscule size. Lastly, the cell membrane serves as the outer boundary of the cell, clearly separating it from adjacent cells, which are marked by distinct lines. This illustrates how animal cells appear under a light microscope. When examining the electron micrograph of an animal cell, we see a black and white image highlighting various structures. The cell membrane serves as the outer boundary of the cell. The large nucleus is prominently visible, along with the coiled structures found within it. Additionally, the mitochondria are clearly defined in this image, and we can also observe ribosomes in the electron micrograph. Finally, the cytoplasm contains various organelles, providing insight into the cell's internal environment. This illustrates how an animal cell appears under an electron microscope.